In this video, we will discuss about a cell membrane, structure of a cell membrane, and function of a cell membrane. This is the diagram of a cell membrane in which carbohydrate, protein, and lipids, intrinsic protein, extrinsic protein, and other conjugated molecules are given. This is another label diagram of a plasma membrane. What is a plasma membrane? Plasma membrane or cell membrane is the outermost boundary of the animal cell and inner to cell wall in plant cell. Their thickness is from 5 to 8 nanometer. Sometimes the plasma membrane acts as a selectively permeable membrane, differentially permeable membrane, or semi-permeable membrane and serve as outermost boundary in the animal cell and inner to the plant and inner to the cell wall in plant cell. Allow some substances to cross more easily than other and made up of phospholipid protein and conjugated molecule, separate and protect cell from external environment and provide connecting system between cell and its environment. Other name for plasma membrane is cell membrane, biological membrane, biomembrane, or plasma lemma. How we define plasma membrane? An outermost envelope surrounding the cell that separates and protects the cell from the external environment and provide connecting system between the cell and its environment is called plasma membrane. Then structure of plasma membrane. Structure of the plasma membrane. The plasma membrane basically composed of phospholipids back to base. What is phospholipid? Phospholipids are lipids in which phosphate groups are attached to them is known as phospholipid, or a conjugated molecule of lipid and phosphate group is known as phospholipid. In this diagram, you can see that phospholipid basically composed of head and tail. Head is made up of phosphate while tail is made up of fatty acids. The head portion of the phospholipid, they are hydrophobic. It pair with water, and the tail portion, the tail portion of the phospholipid, they are hydrophobic. In which it pair with water, while the head portion of the phospholipid, they are hydrophilic. It love with water and arranged as a bilayer. You see here in the diagram. Phospholipid, glycolipid, cholesterol are empiphatic lipids. Empiphatic mean that they containing hydrophobic and hydrophilic ends. Empiphatic mean polar hydrophilic head in one side and polar hydrophilic heads on other side, while non-polar hydrophobic tails is present in the middle. The structures of the plasma membrane is given by Danieli and Davison in 1935. He defined that cell membrane is a lipid bilayer sandwiched between two monomolecular protein layer, and it is rejected. Nowadays, the accepted model of the plasma membrane is known as fluid mosaic model, and this model is given by S.J. Singer and G.L. Nicholson in 1972, in which they awarded a Nobel Prize. What is fluid mosaic model? Fluid mosaic model. Cell membrane basically is the membrane is like a sea of lipids. The membrane is like a sea of lipids in which proteins are floating. Is known as fluid mosaic model. Our cell membrane is a lipid bilayer in which proteins are partially embedded like floating icebergs in a sea. The fluid mosaic model describes the plasma membrane as a flexible boundary of the cell. The phospholipids move within the membrane. Our lipid molecules are present in fluid state capable of rotating and moving. The protein occur as a mosaic of discontinuous particles that penetrate deeply into and even through the lipid sheet. Globular protein are irregularly embedded in the lipid bilayer. What is membrane protein? There are two types of protein present in the cell membrane: extrinsic protein and intrinsic protein. Extrinsic proteins, these are those proteins which are loosely attached with the membrane surface, located entirely outside of the lipid layer, either on the extracellular or cytoplasmic surface, also called peripheral membrane protein, and their example is cytochrome C of mitochondria and cell surface identity marker antigens is the example of extrinsic protein. Now, what is intrinsic protein? Intrinsic proteins are those proteins which are incorporated, are present within the lipid bilayer and tightly attached to the lipid bilayer and provide channel for the water soluble substances, also called integral membrane protein. And their example is transmembrane protein, transport protein, channel, and permeases are the example of intrinsic membrane protein. In this diagram, you can see the intrinsic protein, extrinsic protein, other conjugated molecules of the plasma membrane. Protein in which within the membrane, non-polar amino acids are hydrophobic. These are the non-polar amino acids which are hydrophobic, and those amino acids which are hydrophilic, they are present or extend into the extracellular or into the cytosol region of the cell. The amino acids which are non-polar and hydrophobic, and their example are glycine, alanine, valine, leucine, etc. And the amino acids which are polar and hydrophilic, and their example are serine, threonine, cysteine, tyrosine, asparagine, etc. So the polar amino acids, they are hydrophilic, it love with water. These are the association of various types of protein. That protein, if they are present in single alpha helix, it may be present of multiple alpha helix, and when a protein is present in more complex form, it become ruled up and form beta sheet of the protein. Chemically, the plasma membrane composed of lipid, protein, and carbohydrate. An actual component different from tissue to tissue. That cell membrane or plasma membrane, their component differ from tissue to tissue. Lipids of cell membrane are either phospholipid, glycolipid, sterol, and cholesterol. Why carbohydrates are not inserted into the biological membrane? These are one of the interesting questions which arise in the mind of human beings. That carbohydrates are not attached into the are not inserted into the membrane, are not within the membrane. They are too hydrophilic for that. They are attached to proteins, and they are known as glycoproteins. So conjugated molecule of protein with carbohydrate is known as glycoprotein. Membrane carbohydrate perform a function of cell-to-cell recognition for the identification of one cell from other type of cell, and it is an antigen and also known as a major histocompatibility complex, and it take part in the process of immune system. These are various types of four major phospholipids found in mammalian plasma membrane. These are many minor phospholipids exist too. Phosphatidyl ethanolamine, phosphatidyl serine, phosphatidyl choline, and sphingomyelin. These are name of various types of phospholipid present in the plasma membrane of mammals. Cholesterol, as we know that these are also a type of lipid present and attached to other lipids or other conjugated molecules, and these are the structure of the cholesterol. You see here in the plasma membrane, and these are the structure of cholesterol. This is another extensive and label diagram of plasma membrane. You can read it and you can understand it very easily. Movement across the cell membrane. The from outside to inside transporter, enzyme activity, cell surface receptor, cell surface identity marker, cell adhesion attachment to the cytoskeleton. There are various types of functions performed by the protein present in the plasma membrane. Transport means the transfer of any material from one place to another is known as transport. So transport protein are the protein that allow needed substances or waste material to move through the plasma membrane. Equaporin, basically water move rapidly into and out of the cells. So evidence suggests that there were water channel, and these are discovered by Peter Igri of the John Hopkins University and Redrick McNunn of Rockefeller Institute, that certain channels, water channel, will be present within the protein of the plasma membrane. What is diffusion? The transport of material from higher concentration through lower concentration is known as a diffusion. uh, as I have told that in the diffusion, the transport of material from high to low concentration, and there will be no use of ATP energy. No energy is needed, and this type of transport is also known as a passive transport. Then what is the difference between diffusion and osmosis? The main difference between the diffusion and osmosis that a membrane is present between the in osmosis, while a membrane is absent in diffusion. While similarity in between them, that these are both are known as passive transport, no energy are used, and from high to low concentration the material are transported. Various types of molecule can get thoroughly directed. For example, various types of fats and other lipid, they easily pass through the lipid bilayer, and which type of molecule which cannot get through directly, these are polar molecule, water, iron, salts, ammonia, and other large molecule starches and protein that does not directly pass through the plasma membrane. Channels through the cell membrane. Membrane becomes semi-permeable with protein channel. Specific channel allows specific material across a cell membrane. Osmosis. As I have told that osmosis can be defined the transport of material from a region of higher concentration through a region of less concentration through a selectively permeable membrane is known as osmosis. Then if we arrange the cells on three types of solution: hypotonic solution, isotonic solution and hypertonic solution. In isotonic solution, the water move from the environment into the cell and the cell will become turgid. In isotonic solution, in which the solution within the cells as well as outside the cell, they are the same. If, for example, two water molecule enter into the cell and two water molecule leave the cells, and then this type of solution is known as isotonic and the cell will be known as placid. And if we keep the cell in hypertonic solution, for example, salt water, the water will move from the cell into the environment and the cell will become plasmolized. And here the cytoplasm becomes shrink. This type of a cell is known as plasmolized cell. Phasio transport, as the name indicates, the transfer of material from higher concentration to low concentration without the use of energy is known as phasio transport. So there are various types of when proteins are involved for their transport from higher to low, then this type of transport is known as facilitated diffusion. The difference between simple diffusion and facilitated diffusion, that facilitated diffusion is performed by the protein present within the plasma membrane. And here with the help of facilitated diffusion, so no energy or ATP are used. So various types of protein is responsible to perform the process of facilitated diffusion, and phasio transport takes place with the help of protein. What is active transport? The transport of material from low concentration to high concentration through a concentration gradient with the use of energy is known as active transport. Here, ATP energy is used in the material from low concentration to higher concentration, or the substance from low to high is transported with the use of ATP energy. Then it is known as active transport. So here you can see here in the diagram that ATP energy is basically used in this process, and this type of transport of material is known as active transport. Antiport is the various types of there are various types of active transport. For example, SEM transport and antiport. The SEM port is a type of active transport in which two material cross the plasma membrane and enter into the cell. Then this type of transport is known as SEM port, or SEM mean together. And antiport, when one material enter into the cell, another material leave the cells against the concentration gradient with the use of energy is known as a antiport. So these are basically the summary for transport, that simple diffusion, here facilitated diffusion which takes place with the help of protein, active transport here with the use of ATP from low to high concentration, the material transfer is known as active transport. Transport summary. Simple diffusion, which is clearly indicated in the diagram, facilitated diffusion takes place with the help of a protein, and active transport, as I have told, it takes place from a low concentration to high concentration with the use of ATP energy. How about a large molecule? Moving large molecule into and out of the cell through vesicle and vacuole, endocytosis. Endocytosis mean the material enter into the cell. Endo mean end and cytosis mean the cells. The entrance of material into the cell is known as endocytosis. And there are two types of endocytosis. Phagocytosis basically mean the eating of the cell, and panocytosis basically mean drinking of the cell. And exocytosis mean living of material from the cell to outside environment is known as exocytosis. Exo mean exit and cytosis mean cell. So the transfer of material from the cell into outside environment is known as exocytosis. As I have told and I am explained the endocytosis, that phagocytosis is basically the eating of the cell. Here engulf solid particle, and panocytosis here engulf liquid material. Transport of large particle. So endocytosis is a process by which a cell surround and take in material from its environment is known as endocytosis, clearly here in the diagram. And transport of a large particle from the cell into outside environment is known as exocytosis. So exocytosis is the expulsion or release or secretion of material from a cell. If you like the video, please share, watch and subscribe.